Hello, everyone. We're pleased to have you all join us. I'm Lauren Cossey, Scientific Account Manager at CDD, and I'd like to welcome you all to our Collaborative Drug Discovery Quarterly Product Update webinar focused on our new AI Plus module and our enhanced curves capabilities. So before we dive into these exciting updates we have for you, I'd like to quickly go over a few housekeeping items. For any product-specific questions, please use the Q&A tab in the toolbar. This will really help us address your inquiries efficiently during our dedicated Q&A session, as well as answer any quick ones as we go. For all other questions, comments, or general discussion, please use the regular chat box tab in the toolbar, and we do encourage your active participation throughout the webinar. If you would like to request a one-on-one customized demo for any of these features, we'll have a poll to easily request one before the Q&A segment. So for today's updates in regards to the new AI Plus module, we'll be going over seamless protein structure prediction directly within CDD Vault, integrated molecular docking simulations, visualization tools for structure-based drug discovery, and the NVIDIA BioNeMo platform integration requiring no setup. In regards to our curve module enhancements, we'll be going over exponential decay and scatter plot options with auto-calculated parameters, line graph curve types for visualizing relationships between variables, exponential accumulation and decay fit for biophasic data analysis, and biophasic Michaelis-Menten curve fitting capabilities. However, before we get into these updates, if you're interested in automation and inventory management capabilities, I do recommend you check out our Q1 2025 product update webinar recording on inventory and automation, which is available on our website. These modules have been developed based on user feedback and research requirements with the goal of streamlining your analytical workflows and accelerating structure-based drug discovery processes. And your feedback continues to be invaluable to us, so we encourage you to engage actively throughout the presentation. Thank you once again for joining us today. Let's begin our product update journey with our AI Plus module integration.
Hello, everyone. My name is Salima. I'm Director of Business Development here at CDD. Today, it's my pleasure to introduce our newest AI Plus module, featuring the integration of AlphaFold 2 and DiffDock via NVIDIA's BioNeMo. For researchers, especially engaged in structure-based drug discovery, these tools eliminate the traditional barriers between protein structure prediction, molecular docking, and data management. Let me highlight a couple of key areas where our AI Plus module is going to transform your research. First, we have integrated AlphaFold 2 for protein structure prediction. Now you can generate 3D protein models directly from sequences within your vault. Visualize predicted structures with confidence metrics, store and organize these models alongside your experimental data, and eliminate the need for external platforms or computational infrastructure. Second, we have incorporated DiffDock for molecular docking. Now you are going to perform docking simulations between small molecules and target proteins, view multiple binding poses with scoring metrics, and compare docking results across your compound series, and generate structure activity insights directly within your existing workflows. And thirdly, we have streamlined this whole entire process through seamless integration. Now you are able to move from sequence to structure to docking in a single environment. You can link computational predictions with experimental assay data. Needless to say that you can share your results securely with collaborators and maintain complete traceability throughout your research pipeline. So this integration represents a significant advancement in making computational tools accessible to every single scientist, regardless of your computational expertise. Thanks to our partnership with NVIDIA BioNeMo, these sophisticated AI models run in the cloud with no setup or maintenance required from your side. So now I'm going to hand it over to Susanna, who will demonstrate these features in action right in CDD Vault. Susanna, over to you.
Hi, everyone. Welcome to this webinar. My name is Susanna Tomasiu. I'm a Senior Application Scientist at CDD, and today I will be showing our brand new AI Plus module, which integrates protein structures into CDD Vault. This is an integration of NVIDIA BioNeMo microservices for AlphaFold 2 and DiffDock models into CDD Vault. AlphaFold 2 predicts protein structures with remarkable accuracy using deep learning, while DiffDock leverages diffusion models to predict protein ligand binding poses without the prior knowledge of the binding site. It is easy to set up by adding a readout to any protocol, and the calculation or job is submitted once a readout row is added. So let's take a look at how this is integrated in CDD Vault. Folding and docking models are added as a readout on a new or existing protocol. Let's start with the folding model first. Here I have a protocol with a readout definition, which triggers the AlphaFold 2 calculation. This is a pick list data type with the item yes to trigger the calculation. Then we have a second readout definition to run the actual AlphaFold. The data type is folding, and then in the model, we select AlphaFold 2. In the trigger, we need to select the trigger that we previously created. Now, in the same protocol, we can have another calculation, in this case for ESM fold. We have the trigger also to ESM fold, which is a pick list with the data type yes. And then we have the readout definition in which the data type is folding, the model is ESM fold, and then the trigger is the run ESM fold trigger. Now, to run a calculation, a folding calculation, all we need to do is to go into the Run Data tab. And then here we can either create a new run or select an existing run. And then we need to add a readout. Here, we need to add the ID of the protein amino acid sequence that is registered in CDD Vault, the batch, and then whether we want to trigger the AlphaFold 2 calculation or the SM fold calculation as defined in my protocol. Once we add the readout, this submits the job into CDD Vault and automatically generates all these readouts. So this is a job that has finished already. It's we can see the score, the ID, the job ID, the status, the errors, when the job started, when it finished, and when it was updated. And then this is the actual run AlphaFold output, which is a PDB file. We can double click and this opens the mole viewer in 3D. And here, this is where I can interact with the protein, with the outputs. We can look at the solvent accessible area, the solvent excluded, van der Waals, or the one-sided surface. Let's now take a look at the DiffDock calculation. Here I have a protocol, and similarly, I have two readout definitions. One is the readout definition to trigger the DiffDock calculation, which has the item yes. And then we have a second readout definition where we have the data type is docking. We've given the name, and I like to add the name of the target in the readout and the DiffDock model and the trigger that we created previously. We also need to upload the PDB structure of the target. This can either be the crystal structure or the output of AlphaFold, which is also a PDB. Now, to run a calculation, a ligand docking calculation, again, we go into the Run Data tab. You can either create a new run or add a readout into an existing run. Let's add a readout. Here, I'm going to add the molecule ID of the ligand that I want to dock. And then I select yes, because I want to trigger the docking of this ligand into GLP-1R docking. Let's add this readout. And when we do that, the job has now been submitted, as we see here. And then this should take just a couple of minutes. And then it will be populated with the docking score and also the output of the ligand at the binding site of the docking. So we can use this mole viewer in 3D to actually look at the ligand in the binding site. Okay, so the examples that we saw here, a couple of examples are for running either a folding or a docking manually via the interface. Now, if we want to dock a list of ligands into multiple targets, or if we want to predict the protein structure of multiple proteins, then we can have a protocol which combines the folding and the docking. And in this case, I have the docking to different targets. And then we can have an Excel sheet similar to this one, where we have the molecule batch IDs of the ligands that I want to dock, and yes, to trigger the docking calculations. And then here I have the molecule batch IDs of the amino acid sequences of the proteins that I want to predict the folding. And also here, yes, because I want to run the folding in the AlphaFold 2 and ESM fold. So then we take this into the import data tab. We map the fields as we usually do when we are uploading the data, and all these calculations are submitted automatically. Please note that when a calculation is running, you'll see this banner at the top of the screen. So you'll be able to see the folding results and docking results in a search results table like this one here, where we can see the amino acids sequence registered in CDD Vault and the folding results with AlphaFold and the ESM fold. Also, we can see in the same table the ligands that are docked to GLP1R protein. We can also see just the ligands that are docked into a particular target. And here we can see a list of all the ligands along with the docking scores. We can click on a particular docking, and then we can view the ligand at the binding sites. And I always find this option here to show the residue labels very useful, as we can interact with the viewer and also see which residues are close and making contact with the ligand. Also, we can look at the compound page. As we see here, we have the ligand, the compound, and then under the protocols, you'll see all your AI Plus results in this view. And just to summarize, the typical CDD Vault workflow involves the registration of compounds or biological entities, the registration of assay data, and then analyzing, reporting, searching, and visualizing the data. This covers the make, test, and analyze of the DMTA cycle. The AI and AI Plus workflow includes having a hit or a lead compound, which is registered in CDD Vault, and then using the AI module to find similar compounds with the deep learning and provide bioisosteer suggestions. Then we register those similar structures or bioisosteers into CDD Vault, and then we run AI Plus module to dock those ligands to the targets. Therefore, the new CDD Vault workflow includes the design of compounds with the AI and AI Plus tools, and that closes the DMTA cycle. And that's it for today. Thank you for your attention, and let us know if you have any questions or if you'd like to try this new AI Plus module. Thank you.
Hello, everyone. I'm Ralph Foster, Director of Business Development at CDD. Today, I'm excited to introduce our significantly enhanced curve module designed to help you efficiently analyze and visualize complex biological data. For research teams working across diverse assay types, these updates provide powerful new mathematical models and visualization options that are essential for deriving meaningful insights from your experimental data. Let me highlight 4 key areas where we've expanded the module's capabilities. First, we've added exponential decay and scatterplot options. Now you can apply first-order kinetics to microsomal stability assays, visualize data relationships without forcing mathematical models, calculate elimination rate constants automatically, and customize visualization parameters for clarity and precision. Second, we've incorporated line graph curve type, the ability to connect discrete data points for trend visualization, compare related values across multiple variables, generate clear presentation for time course experiments, and simplify data interpretation with auto-calculated metrics. Third, we've developed exponential accumulation and decay fitting. Now you're able to model biphasic cellular responses with proliferation and apoptosis phases, calculate both K1 and K2 rate constants automatically, generate half-life parameters for complex biological systems, and apply custom calculations using auto-generated parameters. And lastly, we've added specialized curve types for advanced applications, biphasic curve fitting for complex cellular responses, PK and Nicholas-Menten KD fitting with parameters calculation, flexible configuration options for diverse experimental contexts, and integrated calculation for comparative analysis. These enhancements allow you to apply the right mathematical models to your specific assay types, whether you're working with enzyme kinetics, cellular viability, microsomal stability, or other specialized application. Now I'll hand it over to Mitchell, who will demonstrate these features in action, showing you exactly how to apply these new curve fitting options to real experimental data within CDD Vault. Mitchell, over to you for the demo.
Hey everyone, my name's Mitchell Buckley. I'm one of the application scientists here at CDD Vault, and today I'll be covering some of the newest features within the Curves module, which is a great place for users to streamline data analytics, all within the safety and the convenience of CDD Vault. Once Curves has been enabled within a particular vault, the Vault administrators can navigate to the Settings tab, Vault Settings, and then they'll find the Plots sub-tab here, where we have all of the different curve fits offered within the Curves module. Within this window, the Vault admins will be able to select which curve fits they want users in their Vault to be able to apply to data, and this, of course, will ensure that only the correct curve fits will be able to be used. The first example we'll cover is the exponential accumulation and decay curve fit. And this model is a process that's comprised of two phases, including the accumulation and, of course, the decay phase. Each will have its own set of calculated readouts and fit parameters. In this particular case, we're looking at cellular proliferation and apoptosis assay. And of course, we have our curve fit in the center of the screen here. We have an intercept calculated on the right-hand side of the peak, representing our T50. And then we have a series of auto-calculated readouts on the right-hand side of this table, including things like the AUC observed, our min and max measured, our R squared, so the quality of this particular curve fit, as well as the accumulation rate constant, or rate constant 1, and the decay rate constant, or rate constant 2 here. All of these parameters, any calculated outputs, can be referenced in further custom calculations, and that will be true for all of the curve fits that we cover today. In the next example, we'll cover the exponential decay curve fit, outlining how this can be applied to a microsomal stability assay or any of these ADME assays that you might be running. In this particular protocol here, we have a very simple time point of minutes, as well as some raw data coming off of our LCMS instrument in this experiment. The first plot type that we're showing here is an exponential decay curve fit of these LCMS counts versus our time course. And in this particular report, I've defined the decay rate constant here. Moving forward, I've also defined a custom calculation to calculate the available substrate remaining over this time course. And of course, I have plotted that here using that same exponential decay curve fit. I've then defined another custom calculation within this protocol here to take the natural log of our remaining substrate. And then I've used a second curve fit, our linear fit, to fit that log transformed data here versus time. Now, the great thing about this is that I can then use our auto-calculated slope value in our half-life calculation. And really, in this example, we've stitched together a few different curve fits, a few different custom calculations, and it's very representative of the power of the curves module itself. The next example we'll cover is the biphasic curve type, and this is a double sigmoid curve and is typically representative of processes that are comprised of two phases. And so in this case, we'll consider some dual mechanism of action, agonist compounds, or maybe this could be applied to a combination of therapies. The inputs for this particular protocol here are concentration and response, a normalized readout across a few different cell lines that we've tested against. And so, of course, we'll calculate an overall IC50 value for this curve type. We'll calculate the log center 1 and center 2, which would be representative of your left and right IC50 values. Of course, we'll calculate the slope for each of those different phases of the curve fit, as well as other custom calculated parameters. And any auto-calculated parameters can be found within the Customize Your Report menu. Moving to the next example, we'll cover enzyme kinetics with the Michaelis-Menten equation to calculate Kn. And so, of course, this is the rate equation for a one substrate enzyme catalyzed reaction. And in this particular example here, our inputs are substrate concentration and reaction rate. And we'll have a series of calculated properties for this specific curve type, as well as, of course, rendering this specific plot. Of note, of course, we are calculating Km. We'll have an AUC calculation that is automatic, as well as Vmax. And so we know that this is a very important equation to many of our customers who are working in the enzyme kinetic space. We'll next move into the pharmacokinetic space and focus on our IV and PO dosing curve fits. So this is a really exciting development for the CDD team and, of course, all of our customers. In this particular protocol here, we've defined two inputs for our time point and our plasma concentration, and we've also specified two different conditions for our dose as well as our species so that we can aggregate all of our calculations correctly. In the particular report that we're looking at here, I've toggled on our AUC to infinity calculation, our elimination rate constant, as well as our half-life using that elimination rate constant. Our plot will be found on the right-hand side of this table, and of course, we'll aggregate over those calculations that we defined. Within the Customize Report window, if I open this up, we'll be able to see all of the auto-calculated parameters for this specific IV protocol. And so you'll notice we do offer three different AUC calculations, this first one being the AUC observed, so just for our time points that we've specified, as well as a handful of other highly useful calculations. All of these auto-calculated parameters, of course, can be referenced in further calculations. So you may want to calculate clearance or volume of distribution. All of that can be conducted as a custom calculation. In addition to the IV dosing protocol, we now offer as well a PO dosing protocol. And so in a very similar fashion here, we're looking at plasma concentration versus time point over a few different species, including mouse and dog here. And you'll notice that within this particular report, we've pulled in some of those same auto-calculated parameters like our AUC. We've calculated a Cmax here for both our mouse and dog conditions. And we also have turned on here our accumulation rate constant, as well as our elimination rate constant moving to the right-hand side of the table. In addition, we have calculated our half-life based on those elimination rate constants, as well as the Tmax for this particular plot. And one of the upcoming features that we will be releasing soon will be the ability to toggle our y-axes into log scale. And so, of course, for pharmacokinetic purposes, having the ability to view those semi-log plots will be quite important. And finally, moving on to our last example, We'll cover the line graph and the scatter plot. And so, of course, these two models will have no mathematical fit, but is a really great tool for visualizing data, such as the example that we have here. So maybe we're running a particular in vivo study looking at the body weight of an animal. And so here we have body weight versus time point, and we are able to render those plots and view those plots, even though there may not be necessarily a mathematical correlation between these two variables. And with that, I thank you for your time. Please reach out if you do have any questions. And of course, we'll be happy to set up more time to discuss. Thank you.
Thank you for a great demonstration of our enhanced platform features, and a big thank you to all of our speakers today for their excellent presentations on our latest AI Plus and Curve updates. And I'd also like to thank all of you in the audience for your attention and engagement throughout this webinar. Your interest and participation are what really drive us to continuously improve our products. So before we move on to our Q&A session, I'd like to draw your attention to a poll that's now appearing on your screen. If any of these module add-ons we've discussed today look interesting to you, please take a moment to vote. You can indicate whether you'd like a customized demo, or if you'd prefer us to send you some resources to learn more about these module add-ons. And your feedback here will really help us tailor our follow-up to best meet your needs. So now let's transition to our Q&A panel. We've really received some great questions in the Q&A box throughout the presentation, and we're excited to address them. So for those who haven't had a chance to ask questions yet, feel free to submit them now in the Q&A tab, and we'll do our best to answer as many as possible in the time that we have remaining. So let's begin with our first question.